Okay, perfect. The session is now recording. Good morning to you, dear colleagues, and good morning, Dr. Vincent. Thank you so much for honoring our invite to giving this presentation on the topic Introduction to Research Methodology. And you're very welcome to the Research Capacity Development Unit at the University of Johannesburg. My name is Lawrence, and I'm delighted to be your moderator for today's workshop. I'd like to start off by thanking all the attendees for taking the time to join and also for thanking our presenter, Dr. Vincent, for taking his time to preparing this presentation. Before introducing Dr. Vincent to you, I would like to go over a few housekeeping rules, of which I would like to think most of the attendees are aware of. If you do have any question during the session, please type your question in the chat box, or you may raise your virtual hand and we may indicate to the presenter to address your question. And as I've noted in the beginning that this section is being recorded and the recording of the session and the presentation slides will be posted on our model communities by the end of this week. Please note that I will also be posting a feedback survey in the chat session. By also filling out this feedback survey, this ensures that you get a certificate of attendance. And also we use this for our reporting. And also please to provide some kind words to our facilitator for the hard work. Okay, before I hand over to Dr. Vincent, please allow me to read over Dr. Vincent's bio. Dr. Vincent Abakachi is a postdoctoral research fellow at the University of Johannesburg, an expert in computational drug discovery and medicinal chemistry with a PhD in pharmaceutical chemistry. His work focuses on molecular modeling, virtual screening, and molecular dynamics simulations using tools such as the Scrivener, Maestro, and Amber, and Autodoc. Dr. Vincent is published widely in peer-reviewed journals and actively mentors postgraduate students in both theoretical and practical aspects of research. His interdisciplinary experience and passion for capacity building make him an engaging voice in research methodology and scientific development. Without any further ado, please allow me to hand over to Dr. Vincent. Thank you so much.
Thank you so much for introducing me. I'm happy to be a facilitator for this section, and I believe that by the end we should be able to learn one or two things. Thank you for having me, and thanks for everyone who has joined. I'll be presenting on the introduction to research methodology, and so I will be sharing my slide so that you guys can see. Can you see my slide, please?
Yes, perfect. I can see your slides. Please indicate with a thumbs up if you can see Dr. Vincent's slides.
Is everyone
Okay,
fine?
perfect. Yes, I think we can all see your slides. You may proceed, Doc. Thank
Okay,
you.
then. All right, then. Thank you.
Sorry, Doc, just to interject, would you like questions during the session or at the end
Yeah,
of the
maybe
session?
at the end of the session.
At the end of the session.
Yes, yes. And
Oh, okay, no.
if should I deem it fit, I will ask if I want any question within the section.
Okay, not a problem. Okay, you may proceed. Thank you.
Yes. All right, then. Thank you, everyone, for being here. Good morning once again, everyone. Thank you for joining today's workshop on Introduction to Research Methodology. I'm excited to share this foundation now topic with you guys. And I believe everyone will be able to pick one or two things, just like I said earlier on. My background and experience with research methodology is I've been a PhD student in medicinal chemistry, stroke pharmaceutical chemistry, and I have about 10 years experience in both pharmaceutical chemistry and computational chemistry. I believe by the end of this hour, you'll be able to understand research methodology from my own experiences that I've garnered over the years, the key components and how to apply to your academic projects. Whether you are writing a thesis, publishing a paper, or applying for a grant, a solid methodology is critical. Methodology is very important in anything that we want to do. So we want to be able to see and understand it. So I believe this section will be able to help as many of us as in the training and even those who watch it later on. So let's go. We'll go into the next slide to be able to give you guys the agenda of today's topic. All right, the agenda will be I'll be talking from introduction, what is research, what is research methodology, key components, and practical applications using my own work that I've been, one of my papers that I published. I will use it as one of the practical applications in understanding this research methodology. And then we'll go for a QA section and then the conclusion. So we'll see how it goes. What is research? It's very important for us to be able to understand what is research before we go to the methodology. So the term originates, just as I've written there, from the concept of research, meaning to search again, explore thoroughly through repeated investigation. The question we need to ask is, what are we searching for? And so what are we researching? And the simple answer is we are searching for answers or solutions to research problems. And so what are we searching or researching is we are trying to find a solution to problems in our society. When you hear the word research, what should come into your mind in the repetition of the search is repetition of the search. And therefore, we are repeating, searching, and then researching for answers and solutions. So answering or researching problems, and that should tell you something about what you are trying to do. There is no time that we shall say we have researched enough unless we close the word research, because in real sense, the word research means it continues throughout our lifetime. It doesn't stop. From you have your own research, another person also will be able to take it off from where you've stopped and then move forward, because our society is going to continue to have one thing or the other that arises. So, and then we have researchers that keep on exploring and searching for solutions to those issues. So we are searching for answers and solutions to research problems. So what are these problems? How do they come about that we need to also know about this? Every aspect of human behavior is tied to some form of challenges. And these challenges are reflected in our society. Every day and day, even you wake up, you have cough, you have one thing or the other ailment, you have abdominal pain. Why are you having it? These are many challenges that we face in our society. As scientists, we always want to find a solution to those things. So when people are having issues like that, they have what to go for, the drugs to take to be able to address it. There have been problems in society. There are problems now, and there will continue to be problems in the future. We cannot rule out that because we are in a world, is a moving world, and even you and I we keep on growing and aging. Our systems change, and those systems change through the environmental factors. Some things will cause one form of illness or one form of discomfort or the other. So as a researcher, you keep on trying to find what is happening in the society. Why are people dying, or why are people sick? Something like that. A research problem represents a gap between existing knowledge in the literature and what the researcher seeks to discover. There is an existing knowledge about what is on ground. For example, during the COVID time, the COVID has also happened before. And so we try to understand what was it previously, and then we try to see how we want to address it now. So researchers come into the space to try to find solutions, seek solutions from where it was before and now what is can we be doing with it, and even in the future. Essentially, it means that the difference between what is already known and what remains to be explored or understand is what research is trying to solve the problem. So research is about mitigating the gap. You need to know the gap between where we are yesterday and where we are now. And that's how we'll be able to identify the problems. Then from there, you will take it off from that to move to the next space. Therefore, the goal of research is to seek answers to these identified problems. So it depends on whatever you see the problems that you want to address as a researcher. Then you explore, you go deeper in reading, in studying, in understanding where it was yesterday. Then you'll be able to take it off from there and add your own by filling the gap. This is what research is all about. So after I've given that preamble, I want us to go into deeper by trying to understand what research methodology is. And just like it's written, research methodology is a systematic process of planning, collecting, analyzing, and interpreting data to address research questions. It's a blueprint of credible research. So it's a systematic framework. You need to know it's a stepwise. You don't just jump the gun and go to conclusion. You must know, why are you doing what you are doing? So the why question come into place. Why do you want to do what you want to do? This brings about the questions that keep on to come up from your mind. For example, in one of the publications we made in my paper on Zandone derivatives as SARS-CoV-2 inhibitor, we use a structural methodology, which is molecular docking and dynamic substitution, to evaluate binding affinity of an ACE2 receptor, just to be able to help us to be able to discover the inhibitor for SARS-CoV-2. So a question arises, then you generate data, then you go to analysis, then you go to conclusion. So research methodology, just like we have is written here, is a systematic framework for addressing research questions. The question that comes to your mind is what we're now trying to go and dig into to get data from. Data is now depending on which area you are coming from, which area of the science you are in, maybe in the synthetic department, or you're in the computational department, just like we are now. So, okay, what am I going to do to generate this data? Okay, first of all, the question is, okay, I want to get, why am I doing, trying to address the COVID issue that is arising? Okay, there are no antiviral drugs at the moment. So the question comes, okay, how do I get the antiviral drugs? What has been there before? Oh, what has been there before is not effective. Oh, okay, so what can I do to get a new one? So these are questions you are trying to address. And from there, you try to want to seek database to be able to get related drugs, or you want to formulate a new drug as a synthetic chemist. You want to formulate a new drug through trying to get features in those new compounds that you have gotten. So this will help you to generate the data. And then once you generate the data, you have a comparison that you do with the analysis where you compare them. So that is the process of the research. So basically, just like I said, it's a systemic process of planning, one, collecting, analyzing, and interpreting the data to address the research question that you all initiated from the beginning. So this is the blueprint of a critical research. So in doing this, we also need to follow it step by step to try to understand there are particular steps you need to follow. Key characteristics of research methodology. You need to know the characteristics that keep you in that boundary to be able to actually, at the end of the day, make your discovery. You'll be able to answer the question that you're doing. You're not just doing it. That's why we said the research is planned. So the characteristics of research methodology, number one, is systemic. You need to have, it's very logical. You need to take it step by step in your approach. So you need to be very careful in showing carefulness in your research. For example, research must be guided by well thought out, rigorous methodology. The research design should be structured to produce results that are as objective, reliable, and unbiased as possible. So in that, it requires you to take step-by-step approach to be able to get it to be systematic. We'll also look at the next point, which is objective. For it to be objective, you minimize biases through standardized method. You minimize biases is you avoid compromising along the line. Sometimes your data will not correlate. It's not giving you what they want. All you need to now do is to go back and try to check your methodology and repeat again. not trying to play with the doctor the data so that it will give you the result in your mind. Meanwhile, it is not so, because it is not going to be reproducible. A research that you carry out must be reproducible by someone else, by another researcher. So, but if you allow biases to come, you breach the standardized method, then it will not be reproducible tomorrow. Now you have caused another problem, because the other researcher will have a confusion on what you're supposed to build on from your own research. So research is a continuous stuff. So you must not stop with you. So that is why you have to be objective to minimize errors in your own research, to be able to give out what you need to give out. So second point is research is reproducible. So allow others to replicate the study. So that's the most important thing about research. Also, you need to have research to be testability. In research is very important. A good research study must have objective and hypotheses that are clear, measurable, testable. If they are too abstract or vague, the strength and validity of the investigation can be compromised. Okay, an example is my own work that I did on COVID study. I standardized the docking to ensure objectivity. The docking is required. I will still be going extensively in that. You may not understand what we mean by the docking. It's just we have a library of compounds, and we do docking to be able to get the best out of them before we go to the next step. So that is part of the methodology we're talking about. So I was being objective, seeing that okay, everybody can also be able to reproduce this docking process that I'm using. Another thing that we need to also note is importance of research characteristics. We have 3 points that I've mentioned there are important of research characteristics. The validity must also be taken into consideration. Validity there means accurate result. Just like as I mentioned earlier, your result must be accurate. Your result must not be doctored. Your result must not be biased. You have to be sure that this result are the result as a result of the outcome of whatever steps you have taken in your research, depending on what kind of research you are running. For example, if you are doing a synthesis, different steps. One of the things I've discovered about some of us, that's why we are trying to make sure we do a thorough speak in this kind of section, so that people will understand the genuinity of why you need to do a thorough work and accurate result, because research is a continuum. So if you give a valid point or valid result, the next person who is going to continue with your research from where you stop will be able to know and follow some of the processes and methodology you have used to take the research to the next level. For example, as a computational chemist, we source for compounds to target specific drugs, for example, sickness or diseases, for example. We may not have taken it to the next step, which is going to in vitro and doing the in vivo if possible. We'll just do the computational work, get this lead compound, and submit it as maybe a published work. Somebody else will take it from there, the medicinal chemist will take it from there and move further to clinical trials if possible. Now, if I have given wrong data, wrong analysis from my own point, the next person who will look at this published work will not be able to proceed with it, because I'm giving him a contrary result from my report. So we need to ensure validity of our research. It needs to be accurate result. It needs to be reliable. The reliability must be so consistent. Your result must be consistent with your result, your report. Credibility, it has to be credible. One of our papers, you can see, as I put there, it has been published because it goes through the peer review, goes through all those processes, and makes the work acceptable. And that is why this work was able to be published, because it went through the following processes of peer review, and was discovered that it was fine and robust, leading to publication. Another key word we need to take note is a key component of research methodology. Number one is research problem. All these we have talked from all my initial, but I'm breaking it down for us to be able to also have a grasp about what we need to identify a problem. In any research, you identify the problem. Once you identify the problem, then you begin to, okay, source for information about this problem. What has been the result of this problem? What has happened yesterday? What I've done so far? How has it impacted the society? How has it been dealt with or treated already? So those are literature studies you need to go for, to search for, to ask for questions. This is where literature come in. So research is not topic driven. Research is problem driven. It's the problem that drives you. And once the problem drives you, you see it begins to open a lot of doors for you to be able to ask questions. And once you begin to ask questions, it begins to open up a bigger world. Oh, okay, this is what I need to do. Oh, this is what I need to do. Then begin to bring more concept into your mind on what you want to do. Identify the problem. For example, in the work where we publish that I'm using as a benchmark in this presentation, lack of effective COVID inhibitor was what the first question that came to the problem that came. There is no effective drug. We only have antiviral. We don't have it. So what the question now arises, what can we do? How do we source out this? This is what led us to look for compounds related that have antiviral relations, which are some natural products, xanthone derivatives. After we made a study search and we were able to find that these compounds have antiviral, potentials. So we decided to want to subject them to see if we can get an effective inhibitor from there. So that's number one. So we formulate questions in that direction. So from there, we are able to, it now led us to all our questions about xanthone derivatives. It now led us to be able to formulate other questions that led the study to be able to get robust. And then we now decide, okay, we will narrow these compounds that we have found. We'll narrow it to be able to get our lead compounds. The second key components in research methodology is literature review. I've mentioned it earlier in the work. You need to be purpose, you need to have steps, and you have tools that you need to find out. In our own case, we use different kind of search database to be able to search for literatures related to COVID that people have worked with. So these helped us to be able to read, understand how far it has been done, how many people have worked on it, what is left, because we will still keep on to look for a particular inhibitor that will be able to address this. So since it has not already been gotten. So for our SARS-CoV study, for example, we reviewed pro works on ACE2 inhibitor, finding limited data on xanthone. You see, finding limited data on xanthone derivative compounds that is associated with COVID. This was one of the things that pushed us to want to focus on that particular project. So we justify our focus. Okay, it now took us the next step, which is the tools that we're going to use to search for these particular important the compounds that are related to xanthone. We use different systems like Google Scholar to do our literature search. We use PubChem to search for related compounds. And so there are millions of, it's a database, we have millions of compounds there who we're able to search out these compounds because a lot have been done and have been deposited into this database. All we needed to do is to bring out this particular compound and do studies on them to be able to isolate them out and say this compound is good for this particular question that we want to address. In our own case, maybe SARS-CoV inhibitor. The next key component in research methodology is research design. Please, I hope everyone is going on with me. I hope I've just been talking. I hope people are getting me. I want to share a thumbs up or a show of hand that I'm making a point. And some people are getting me. I'm not just running and speaking.
Yes,
a show
but
of
I
hands.
think we're on the same
Okay.
boat. I think we can proceed. There's no questions at this
Okay then.
moment.
All right.
I see thumbs up, so I think we can continue.
All right then. Thank you. So,
Okay, thank you.
so one of the key components again is research design. Research. These are all the company. One of the company we talked about the literature. Now I'm talking about the research design. One of the aspects is data type. Research design. We have two major types of research. quantitative and qualitative. In our own case, I always prefer us using both because you use different quantitative like docking scores database to try to get our data. Then we analyze them, we interpret them quantitatively. So we mix both in our own case to try to do our research. We have the objective, which is measured quantitatively. We explore the meaning. This brings about the analysis. Okay, the meaning of what we have, gotten through the quantitative measure. So in our own case, is both we combine. Most of the time, research is combining of both quantitative and qualitative. So quantitative is numerical data, example, docking scores. Qualitative is narrative data. Kind of, you're analyzing, what you have gotten from your quantitative. So mix both, combining both is what I always subscribe for doing research. Our study was, one of the examples in our study was using molecular docking and dynamics to measure binding energies of xanthone derivatives, targeting ACE2 protein. All right. So that was one of our own, my own example of a quantitative. Quantitative, we are using a tools, which is docking score, to be able to analyze. The last one in the components is the data collection and analysis. The method is a computational. In our own case, I'm using my own case of my work that I've done as an example. Method, sampling, analysis, and the ethics is what encompass data collection and analysis. So the method, in our own case, in one of the works we worked on SARS-CoV, computational simulation was carried out, docking was done, and then we did the binding energy calculation. So these are the methods that caused us to arrive at our final result. We did sampling. Sampling was what we used the database I earlier on talked about, PubChem, to try to dig out similar compounds that are called xanthone. Since we have identified xanthone as an antiviral related compounds, so we use a database to bring out a number of compounds. So we brought about a lot of compounds, about 200 compounds. And these 200 compounds, we sampled them out. Sampling them resulted for us to get 91 good ones that are, giving us good scores because we rank them based on scores. So then we analyze these docking scores, and then based on the docking score, binding energies, and the MMGBSA analysis that we got. So based on that, we were able to isolate the lead compound from this. These lead compounds are, for example, then we did the ethics. We ensured transparency by documenting all the computational parameters. In our studies, we docked 91 compounds, selected xanthone 71. That's what we are seeing. The binding interposes I used to show you on the diagram as a diagram, just to be able to show you one of the compounds that shows good activity. And then for their superior binding target and analyzed with MMGBSA. So these are one of the things that we did for our own compound. So data collection and analysis, I've used my own one of my own projects to be able to give you details of going from the methodology, sampling, the analysis, then the ethics. So to cap up what we have been saying, practically, I'm going to focus more. Let's drive into my own study as a case study. The title of the work was In Silico Exploration of Natural Xanthone Derivative as Potential Inhibitors of SARS-CoV-2 Replication and Cellular Entry. So the research question from the beginning was, can xanthone derivative inhibit SARS-CoV entry via ACE2? So this was the research question. And then when we were able to answer this question, we went to the next step, which was the methodology. Without answering this question, you cannot go to the next step. But immediately we're able to answer that question, it not made us to be able to go to the next point, which is the methodology. How do we go about to isolate the right compounds from this xanthone derivative? Because all of them may not be very, very active as required. So, but some of them will have been. So this takes us to the methodology at which we will use to get them. So we start by quantitatively. The molecular docking and the dynamic simulation are the tools we use to be able to see how we'll be able to get through to the right compounds. So we did sampling, we did the analysis, which is the docking, the same I've mentioned earlier on. Then the outcome was the xanthone 71 and xanthone 72 as promising inhibitor, and then we publish it in the Journal of Computational Aided Molecular Design, paving the way for an in vitro validation. Just like I said, research is step by step. We have done ours as computational chemists. We have provided this compound to the research world, and we believe, we can also take it up to the next step. The synthetic chemists, we synthesize this compound. So we have it. When we have it as a synthetic, when the synthetic chemists have it, we take it to the biological lab where we test it and then validate what the computation has given us. Then from there, it will go to the clinical step. So that is how the research goes on. That's why we say research is a continuum. It's not a stop. So research is also based on literature, documented. Research is not a hearsay. It must be documented. You must document your findings and then deposit it, or you have published it so somebody else can run with it. So in rounding up from what I've talked about from the beginning now, a strong methodology is a key to academic success. Without having the right methodology, you will not be able to get to the end of what you want. Methodology is a rationale behind your work in the initial. Then we have methods, the tools that you use to achieve your methodology. So a strong methodology is a key to academic success. Very important to have that. Many people run away from the method at which the processes at which they go through to get things. They just, jump the gun and just get data from nowhere and just write out some things. So you don't even understand when you are reading such kind of publications at the end of the day. So we are in the section for the Q&A. I think I've been able to say some things for one or two questions to be able to come up. So I'll be glad to hear some of the questions. Thank you.
Once again, thank you so much, Dr. Vincent, for the very insightful presentation. A very insightful session on introduction to research methodologies. And I think now it's time for the session, as Dr. Vincent has mentioned, that it's open discussion. So if you have any questions, please raise your hand, or you can either type the question as well in the Q&A session or in the chat box. And Dr. Vincent will be here to facilitate the questions. Also, please note that I have posted the feedback survey in the chat session. Please kindly take a few minutes to fill it in. Also, this ensures that you get a certificate of attendance for this particular workshop by filling in the evaluation form. Thank you so much. You can raise your virtual hand, or you can type the question in the chat box, and Dr. Vincent will be able to address your questions. Thank you so much.
Was he having any question or okay?
Any question at this point?
Okay then.
I see there's a hand raised from you there. I think maybe that's a mistake. Okay. I see there's a hand raised from you again.
Oh, okay. There's another person in I'm using somebody's platform. So that's why maybe the person is also having his platform maybe.
Okay. Or do they want to share something, say something from their side?
Okay, thank you, everyone. I have a question for Dr. Vincent. I just want to ask about the methodology. How should I select an appropriate research method for my study? And what considerations are important when working with a small data set?
Thank you so
Hello.
much for the question.
Thank you for the question. Number one thing is just like we highlighted from most of what we have been saying. How do I choose a method? Now, when you have a data set already, especially when it's even a small data set, the pilot studies, you need to know the limitations. So the first thing you need to do is docking to be able to narrow down your compounds. We don't go to the next step. The docking gives us a score, and then the binding score within that's as a computational chemist. Some of the people in the house may not know what I'm saying, but based on our own research pathway, we start with docking to be able to get the binding nature of those compounds. So once we get the binding nature, it assigns a score. So the most negative score will be what we're going to use, and we have a benchmark. It's either you are using an existing drug which maybe has been failing, so you are trying to make sure your own compound is better than that drug. So if you do the initial docking score, it will evaluate it based on their scores and see if your own compound is better from the score. So if the compounds that are better are moved to the next step, which is the MD. And the MD will help us check the stability of these particular compounds. So once it's able to show the stability, we will now take it off to the next step. So that is how we filter and eventually get the lead compound from that. So that's how you choose your methodology in this area of computational chemistry. I hope I've been able to answer that question. Thank you.
I hope you answered Alex, with that question. I hope you answered there.
Mm.
Okay. Perfect. I do have a question of my own,
Okay.
Dr. Vincent. So my question is, what makes a good research methodology, and how can one figure if a certain research topic you need to use a quantitative method or qualitative method for a particular research topic?
Oh, okay. Thank you for that. There is no research that is not good, because you are seeking knowledge and you're trying to also add knowledge. So in any way that you're going to go to, in the quantitative, just like we've mentioned, is not just carrying out a survey, just like the qualitative. The quantitative, most of the science world, which is in the science field, not in the social sciences. The social sciences, we try to do maybe survey, ask questions, then you try to sample, and then you'll be able to begin to write. So that may be qualitative. But in quantitative, you need to get your data, try to understand from literature what has been done. Then you need to have a method. There are tools that you use to be able to do that, which is a qualitative. So which will lead you to be able to get you the answer of what you are trying to get, the question you're trying to answer from the beginning. So the difference in the two is depends on the area you are. Most of the time in the sciences, we do more of the qualitative, but both is also important in the sciences. But in the social sciences, they do more of the qualitative, where they carry out surveys, ask questions, and take samples from what is happening in the environment to try to get their methodology done. So that's the way I can be able to answer in this area. I hope you're able to get it.
Yes, okay, thank you. Thank you so much, Dr. Vincent, for clarifying on that question. Thank you so much. I see Vitalis, you had your hand up. Did you want to follow up on something there?
Yes, it was a question that I wanted to ask.
Thanks.
to Dr. Vincent. I think that will be last one from me. So I know that publishing in high impact journals within computational chemistry can be quite challenging, and many researchers face multiple rejections along the way. So as an expert in this field, could you share your strategies for successful publishing in top tier journals? And additionally, how do you approach and manage reviewers' feedback and rejections to ultimately achieve these publications? And what are the key practices or mindset you consider essential for navigating the publication process effectively?
Thank you, Vitalis, for this very good question. Because the end goal of every research is to be able to publish it, is to be able to, give a feedback to the research community to be able to see what we have done. Number one, research publication is very important, and so how to manage rejection also is very important. Peer review is on the line. When you submit your work or any work you have submitted for publication to any journal, I will always encourage that you should not worry about the rejection. For example, one of the works we submitted for publication was rejected. This last work I'm sharing with you as a research was rejected 4 places for various reasons. Number one, they felt the COVID kind of research is already saturated. They wrote the reviewer, the facility reviewer was able to return that word that COVID has been flogged. So that's why they don't want it in their journal. They rejected. So 4 places like that before eventually the 5th one was I got the response to under review. And once they carried it under review, they did that. They returned back. The peer review was very important. That is why I don't say if you want to publish any paper, you should feel that rejection is an issue. Rejection is to help you improve that job. So when the rejection return after other, people have reviewed your work, they have critical things they've identified, and then return back to you. You see, the paper will be improved at the end of the day. And then when you are submitting that publication and it's published out for researchers, they'll be able to value that paper because a lot have been able to go into it. It's not you alone, because sometimes you see it from your own view alone. And so others come into the play as researchers and view it in different way. And when you collectively add everybody's view into that work, it becomes richer and it becomes an interesting research work that's been done. So research is something that you don't just say because you want to get it perfect sometimes from the beginning. No, don't worry about that. Do what you can do, put it together, submit it for publication. Then it will return back. Another paper that we had, I'm sorry if I hope I'm, gotten all the questions you have asked. One of the paper was rejected recently. And the idea of that paper was to take up what the synthetic chemist has done and take it to computation. Now, we did that, and the limitations the computational itemized, we itemize it in that work and submit it for publication. Now, when the result will return from the peer review, it says that it was rejected because there was no novelty. 2, there is no new compound was discovered. We are just validating the computational work. So now, from that rejection, what is the issue? We have been able to get, okay. So we needed to show them there's a new compound, even though we have discovered the limitation in this particular compound that the synthetic chemist has done. We need to now take it to the next step by proposing a new compound from the compound the synthetic chemist did, not to just mention the limitations. So now we are not going to throw away this job. We are going to go and improve it by going to do what we call QSAR. It's another tool. QSAR, where we're going to dig deep to search for more compounds, then dock them, just like as I said, the methodology, dock them, check their MD status, and then be able to get a new compound related to the synthesized compound that also shows ADME properties better than the synthesized compounds. Then that is what we'll be able to submit for publication. So it's a process. So as a researcher, never get bad about rejection. Rejection is part of the process to get us to the final step of getting a resound, rich, published work out there. Thank you.
Thank you. Thank you so much, Dr. Vincent, for clarifying on the question. Thank you so much. I hope you are answered there, Vitalis, or if you have any comment or further questions, you can go ahead. I would like to believe you are answered there.
Thank you so much.
Were you saying something?
No. Thank you. It's fine. I'm okay.
Okay, perfect. Okay, is there any other questions from the participants? If you have any other questions or any contribution to make or any comments that you'd like to give out, please kindly raise your hand or you may type a question in the chat box. Since Dr. Vincent is here, he'll be able to indicate your question or rather address your question. So I'll take it with the silence that everybody's answered. I think the presentation was very quite insightful and very clear from Dr. Vincent with the title Introduction to Research Methodology. I hope everybody has taken from this session, and I hope you'll be able to integrate this into your research going forward, whether you're doing a master's or PhD on honors. So thank you so much again to you, Dr. Vincent. I don't know if you'd like to add anything or you'd like to make any further comments before we can end the session.
Thank you so much for the organizers of this. I don't think I have anything to say further. I think we have exhausted the talk so far. Thank you so much, everyone, for listening.
Okay, thank you so much again to all the participants for taking your time to join the session. And thank you so much, Dr. Vincent, for your expertise on this. And we really appreciate you for supporting the Research Capacity Development Unit. And as I've mentioned in the beginning, that I posted the feedback survey in the chat session. So I'd kindly request all the attendees to please fill it in. And also this will ensure that you get a certificate of attendance for this session. Thank you again to all the participants and thank you again to you, Dr. Vincent. So in that regard, I will now be ending this session, if that's fine with you.
Thank you so much. I'm fine with me. Do enjoy the rest of your day. Yes.
Okay, thank you so much. And thank you so much to all the participants again and enjoy the rest of your day further. Thank you so much.
Thank you so much.
Thank you, bye.
Bye-bye.